Good evening everyone. I'm discussing some important PYQ based topics of biochemistry. So first topic is CRISPR-Cas9. So CRISPR-Cas9 is also a recently asked INICET question. It is one of the component of recombinant DNA technology. It's a genome editing tool derived from bacteria, right? It consists of a guide RNA, which is complementary to target DNA sequence, and an endonuclease, which makes a single or a double-stranded break at the target site. Now, imperfectly cut segments are repaired by non-homologous end joining repair mechanism. This is also asked as a PYQ based question. So this is important. So non-homologous end joining repair mechanism is used to repair the imperfectly cut segments in the CRISPR-Cas9. Now, potential applications involving removal of the virulence factors like pathogens, replacing the disease-causing alleles of genes with the healthy variants, like in clinical trials used for sickle cell disease, or specifically targeting the tumor cells. Next topic is PCR, which is very frequently asked and repeated also. So in PCR, we have molecular lab procedure, which is used to amplify or desired fragments of DNA. Now, used as a diagnostic tool for like neonatal HIV detection or herpes encephalitis, mainly PCR has three main steps: denaturation, annealing, and elongation. So it requires, this is also a question, which is what are the requirements of the PCR? We require DNA template, DNA primers, a heat stable DNA polymerase, and a DNTPs, which are deoxynucleotide triphosphates. We heat it at 95 degrees Celsius temperature. Then annealing, we have to cool it at 55 degrees Celsius. And after that, we have to elongation, that is, temperature is increased again to 72 degrees Celsius temperature. Next, the DNA polymerase adds DNTPs to the strands to replicate the sequence after each primer. Heating and cooling continues until the amounts of DNA is sufficient. So there are many variants of PCR like RT-PCR, that is the reverse transcriptase PCR, which is used only for the RNA detection. This is also a PYQ INICET question. Then we have a real-time PCR. Real-time PCR is used for only quantification of DNA. We have one more component that is called as nested PCR. Nested PCR is used when we require or when we use two primer sets. Next topic is elastine. Okay, so elastine, mainly elastin found in skin, lungs, large arteries, elastic ligaments, vocal cords, epiglottis, and ligamentum flavum. A PYQ question, ligamentum flavum contains maximum elastin fibers. It is rich in proline, glycine residues versus hydroxylated residues of collagen. So then cross-linking occurs extracellularly via lysyl oxidase and gives elastin its elastic properties. There is one clinical correlation that alpha-1 antitrypsin deficiency results in unopposed elastase activity, due to which can cause COPD. Now there are two important diseases, Marfan syndrome and homocystinuria. So Marfan syndrome is autosomal dominant disorder, connective tissue which is affecting mainly the skeleton, heart and the eyes. Fibrillin 1 gene defect is seen in this condition, which is also a recently asked question. Fibrillin 1 gene defect is seen in Marfan syndrome. In this, the findings are tall and a long extremities patient with a chest wall deformity, pectus carinatum, pigeon chest or pectus excavatum, hypermobile joints, tapering fingers, arachnodactyly, cystic medial necrosis of aorta, then aortic aneurysm or rupture, the most common cause of death in Marfan syndrome. Mitral valve prolapse, increase the risk of spontaneous pneumothorax. Next disorder is homocystinuria. Homocystinuria occurs mainly because of cystathione beta synthase deficiency, which lead to increase in the homocysteine level. Presentation is similar to Marfan syndrome with pectus deformity, Tall stature. Here we have joint hyperlexity, skin hyperelasticity as well as scoliosis. Homocystinuria is mainly autosomal recessive. IQ level, intellectual disability may be there, and lens dislocation here is downwards. But in Marfan syndrome, the lens dislocation is upwards and temporally. Next disease is Menkes disease, which is a frequently asked question. Recently in INI is also being asked. X-linked recessive disorder, connective tissue disorder hai. Then Menkes protein, ATP7A gene involvement will be there in Menkes disease, while ATP7B gene in Wilson disease. Lead to decrease in activity of lysyl oxidase, which is purely a copper dependent enzyme. Defective collagen cross-linking. This lead to brittle, kinky hair growth and developmental delay. There is also hypotonia and increased risk of cerebral aneurysm. Next topic is cytoskeletal elements. So cytoskeletal elements is also one of the asked question previously. It contains microfilaments, intermediate filaments and microtubules. Microfilaments we have actin and microvilli. Intermediate filaments we have vimentin, desmin, cytokeratin, laminins, glial fibrillary acidic protein, which is a marker for pilocytic astrocytoma, and some neurofilaments. Microtubules, it consists of cilia, flagella, mitotic spindles and centrioles. One more question, paroxysomal defect. Peroxisomes are actually the membrane enclosed organelles which are involved in, these are processes which are involved like oxidation of very long chain fatty acids, alpha oxidation of branch chain fatty acids is purely peroxisomal process. Synthesis of bile acids and plasminogens also occurs in peroxisome. Whenever there is a defect in the peroxisomal oxidation, it leads to three main disorders: Zellweger syndrome, which is there is a PEX gene involvement. Then Refson's disease, when there is defect in the alpha oxidation, it leads to accumulation of phytanic acid. There is phytanol coenzyme A hydroxylase deficiency and adrenoleukodystrophy, in which is there is beta oxidation defect due to mutation in the ABCD1 gene. It leads to accumulation of very long chain fatty acids in the adrenal gland, in white matter of brain and testis. Further important topic is heterochromatin, euchromatin difference. So heterochromatin, as you can see in this electron microscopy, heterochromatin is actually a condensed form and it appears more darker in the electron microscopy like this. This is a heterochromatin which appears, this is a heterochromatin which appears more darker. It is sterically inaccessible, transcriptionally inactive. That is heterochromatin. Euchromatin is less condensed form. Like you can see, this is a euchromatin. Appears lighter on electron microscopy, transcriptionally active and sterically accessible. Now DNA methylation and histone acetylation. DNA methylation, in which it changes of the expression of a DNA segment without changing the sequence. It is involved in many conditions like aging, carcinogenesis, genomic imprinting, transposable elements repression, and X chromosome inactivation, which is called lionization. Now DNA methylation always decreases the transcription rate, very important. Now this is a question asked previously. Methylation within gene promoter CpG islands typically represses silences gene transcription. So CpG methylation makes the DNA mute. Histone acetylation, it increases the transcription. So whenever there is methylation, it will decreases the transcription, it makes the DNA mute, and histone acetylation will increase the transcription. Next topic important is the rate limiting enzymes. So rate limiting enzymes include in the glycolysis with PFK1, phosphofructokinase 1, which is being inhibited by ATP in citrate. Gluconeogenesis, fructose 1,6 bisphosphatase. Then the important ones are glycogen synthase, rate limiting for glycogenesis. Glycogenolysis, glycogen phosphorylase, many time asked question. Glycogen phosphorylase is activated by epinephrine, glucagon, AMP along with calcium. So calcium is also one of the activator of glycogen phosphorylase. Glycogen phosphorylase, the cofactor for glycogen phosphorylase is B6, vitamin B6. HMP shunt, glucose 6 phosphate dehydrogenase, G6PD. Then de novo purine synthesis, PRPP amidotransferase. Urea cycle important, CPS1. Remember, CPS1 is for urea cycle and CPS2 is for the pyrimidine synthesis. Fatty acid synthesis, acetyl coenzyme carboxylase. Fatty acid oxidation requires carnitine acyl transferase, which is CAT1. Ketogenesis, HMG coenzyme synthase, cholesterol, HMG coenzyme reductase. This is also a question asked previously. Ki HMG coenzyme reductase is activated by insulin. There is an anabolic hormone, thyroxine and estrogen, and inhibited by glucagon, cholesterol, as well as cortisol. Cortisol also inhibits HMG coenzyme reductase. Next topic is universal electron acceptor, in which we have NAD, NADP from B3, flavin, FAD from vitamin B2, that is riboflavin. So NADPH is used in anabolic process, like in steroid and fatty acid synthesis. Previously asked question, in fatty acid synthesis, which is the electron acceptor used, which is NADPH. NADPH is a product for HMP shunt. HMP shunt is the main source of production of NADPH. NADPH is used in these processes: anabolic process, respiratory burst, cytochrome P450, a previously asked question, and glutathione reductase. Next important topic is amino acid derivatives. This question is asked many times, especially in the INICT questions and in the match the following type of questions. So phenylalanine tetrahydrobiopterin is required. Tyrosine forms dopamine, then dopamine forms norepinephrine, then epinephrine. For the conversion of norepinephrine to epinephrine requires S-adenosyl methionine. Tryptophan forms niacin and serotonin further form melatonin. Histidine, histamine, glycine forms porphyrin and then heme. Glutamate forms GABA as well as glutathione. Arginine. So arginine forms three structures which include in creatine, urea and nitric oxide. Next important topic is the glycogen and the lysosomal storage disorders. So lysosomal storage disorders, which includes Tay-Sachs disease, in which we can see the cherry red spots on the macula. These are lipid accumulation in the ganglionic cell layer. There is characteristic feature is there is no hepatosplenomegaly in Tay-Sachs disease. Hexazoaminidase A deficiency will be there, and GM2 ganglioside will be accumulated. Fabry disease, alpha galactosidase A deficiency, X-linked recessive. All are autosomal except Fabry disease, which is autosomal, which is X-linked recessive. It includes angiokeratoma, the most characteristic features. Metachromatic leukodystrophy, Krabbe's disease, Gaucher disease, important. In this, the bone marrow examination shows a crumpled tissue paper appearance. The most common lysosomal storage disorders characterized by osteopenia, osteoporosis, avascular necrosis of the femur bone, and the Gaucher cells are characteristic, which include lipid-laden macrophages resembling the crumpled tissue paper. Here we have glucocerebrosidase or beta-glucosidase deficiency. Niemann-Pick disease, neurodegeneration will be there, foam cells and cherry red spot will also be there in Niemann-Pick disease. And sphingomyelinase deficiency. Mucopolysaccharidosis include Hurler and Hunter. So Hurler's mein there is developmental delay, skeletal abnormalities, corneal clouding is a typical feature of Hurler. Alpha L adenose deficiency. So Hurler L, alpha L iduronase deficiency. Hunter syndrome, like hunters, they require a very clear eyes to hunt. So that's why we have a corneal clouding is actually absent in Hunter syndrome. It is X-linked recessive. Further important topic is lipoprotein functions. This table is highly important, high yield table. In this, you should know about chylomicrons. Chylomicron production requires ApoB48. Then chylomicrons delivers the triglycerides to peripheral tissues. VLDL, intermediate density lipoprotein, IDL, which is formed from degradation of VLDL. LDL cholesterol, which is a low density cholesterol, it delivers hepatic cholesterol to peripheral tissues. That's why it's called bad cholesterol. So the characteristic feature is taken up by the target cells via the receptor mediated endocytosis. Then we have HDL cholesterol, which is for reverse cholesterol transport. It acts as a repository for ApoC and ApoE, and alcohol increase the synthesis of HDL. HDL cholesterol requires ApoA1 for the transport. ApoA1 for the synthesis of HDL is required, and ApoB48 for chylomicrons. And for VLDL and LDL, we require ApoB100. A beta lipoproteinemia is an important disorder asked previously. It is an autosomal recessive disorder. There is mutation in gene which encodes for MTP protein, that is microsomal transfer protein. In this, there is chylomicrons, VLDL, LDL are absent. The typical feature lead to falt, malabsorption, steatorrhea, failure to thrive. Also symptoms of retinitis pigmentosa due to vitamin E deficiency. So restriction of long chain fatty acids and large dose of vitamin E is required for the treatment of A beta lipoproteinemia. Next important topic is familial dyslipidemias. This table is high yield for NEAT PG and INICT both. Type 1 hypercholesterolemia, which includes lipoprotein lipase or ApoC2 deficiency, in which there is accumulation of chylomicrons, triglycerides, and cholesterol. Here we have a features of pancreatitis, hepatosplenomegaly, and xanthomas. Type 2 hypercholesterolemia, also called familial hypercholesterolemia, especially type 2A, which have increases LDL and cholesterol. There is defect in the LDL receptors. Here we have cholesterol 300, accelerated atherosclerosis before the age of 20. Tendon xanthomas are also a typical feature of hypercholesterolemia type 2. Beta lipoproteinemia, which is ApoE defect, chylomicron VLDL increase, also called as broad beta disease. Type 4 hypertriglyceridemia, here we have hepatic overproduction of VLDL cholesterol. Here we have hypertriglyceridemia, can cause acute pancreatitis. And also it is resistance related to the insulin resistance. The further important topic is the blood transporters, the glucose transporters. So we have insulin dependent and insulin independent. Insulin dependent glucose transporter is only one, which is GLUT4, very frequently asked question. The locations of GLUT4 are adipose tissue, striated muscles, which exercise can also increase the GLUT4 expression. Insulin independent are further GLUT1, RBCs, brain, cornea, placenta are high. GLUT2 in beta islet cells of pancreas, in GI tract. GLUT3, brain and placenta. GLUT5 is fructose, which is present in spermatocytes in GI tract. Other transporter of glucose is the sodium glucose transporter SGLT1 and SGLT2, which are SGLT2, which is more prominent in the kidneys, Brain prefers to use glucose but may use ketone bodies during starvation. But RBCs utilize only glucose. That is a potential previous year question. RBCs utilize only glucose as they lack mitochondria for aerobic metabolism. Other IMP topics: post translational modifications. What comes under post translational modifications? This is a PYQ question. Post translational modification include phosphorylation, glycosylation, hydroxylation, methylation, acetylation, and ubiquitination. These are involved in the post translational modification, important being the glycosylation. So these are the important topics of previous year questions based, and I have started this series so that more and more questions we can discuss, IMB topics we can discuss. So thank you so much.